And so the first part of my talk, I said I was going to tell you about a specific extracellular regulator. And I've introduced you to LON2. This is work that my postdoc Soprano has done. And that is here in the pathway that I showed you. And this is a family of molecules that's called lipicans. That's, I'll get into the details of that in a little bit. But I want to tell you, why are we interested in this particular extracellular regulator? In humans, there are 6 of them. One of these is known to have a disorder called Simpson-Galabi-Weymouth syndrome. Again, it's an overgrowth syndrome. It's distinct from other overgrowth syndromes that are associated with TGF-beta. It also has an increased incidence of a childhood tumor, Wilms tumor. Also polydactyly, which you saw before in the GDF8 dwarfism mutants. They're considered coarse facial features, as well as cardiovascular defects, which is how I got my American Heart Association grant. Other of those 6 family members also are associated with defects in the cardiovascular system, but they have no idea why. In us, though, the reason why might be because there are other cell signaling molecules that are involved that may be regulated by this family. Lipican has sugar side chains that are post-translationally attached to it. And a lot of things like to bind to these carbohydrate sugar side chains, including TGF-beta. However, in our model system, these other signaling molecules are represented, but they have different phenotypes that are separate from our body size phenotypes. And when we mutate, there are 2 in us, not 6, 2 in the worm, not like the 6 in us. We mutate 1, has no effect on body size, has no effect on our TGF-beta signaling. But the second one, in my mind, is actually the first one. You can see that there's a profound difference in body length. Okay, and with collaborators, we showed that this protein actually binds to the ligand, to TGF-beta, which gives it to negatively regulate body size. So what is this family of molecules? It is, I'm just showing you a nice little stick graph here to keep things simple. It has a signal sequence at the N-terminus because it's an extracellular protein. And so it needs to get out of the cell, it needs that signal sequence. Okay. It has 14 conserved cysteines, which will disulfide bond and give it a nice tertiary structure through most of it. It has sugar attachment sites. This is also called glycosaminoglycan, or GAG. Okay, so if I accidentally say GAG instead of sugar or carbohydrate, you know what I mean. Okay. And they all have these, a variable number. And then they also have a link, a post-translational modification, again, that links this to the membrane on the outside of the cell. And that is called a GPI, or glycosyl-phosphatidyl inositol. Okay, GPI, link. I'm not going to test you on this at the end. Okay, yeah, take a picture, okay. And then some of them, this is not shared in all of this protein family, but this is what makes this a family of and some of them also contain a furin protease site. So this protein will be cleaved into 2 pieces at that site. A 3D-dimensional structure was found on 2 of these a couple of years ago, just a couple of years ago. And it looks largely like this. Remember those disulfide cysteine bonds? You got in LON2, I've represented the sugars like this, the GPI anchor. I just cut and pasted just to give me something to hang these mutations that I'm about to tell you about on, so you can see what's going on. And then there's this protease site right here that separates off these 2 alpha helices and this unstructured alpha helices with this unstructured region, which contains the sugar side chains and the link to the membrane. Got it? Okay. Nodding your head keeps the blood flowing. Okay. So we got the protein. If it's right. This is actually the RGD and not the RXXR. The RGD is not found in all proteins, but this is a protein-protein interaction motif. So we might talk to what its function is. So I'm a geneticist. What do I like to do? And what can I do with my transgenic powers with this model system? Come on. We got the furin cleavage site, we got the sugar attachment. Okay. We got the GPI membrane attachment. Okay. I can address these questions using my tools at hand. I can ask, how can glypicads inhibit TGF-beta signaling, which I already showed. It binds to double one somehow, right? But how? And what domains of the glypicads are actually functionally important? The reason to ask this question, besides the intellectual interest, and can we solve it? Yes, we can, is that these could provide druggable targets. Okay? So this is what we can do in my system. We can take a length of DNA that encodes the LON2. That's sarcoglypican. Remember, knock it out, the animals become long. LON2. What we did is we added a sequence encoding green fluorescent protein. Okay? So, if it works, this protein is going to fluoresce green. We put it into a plasmid with a marker, and we inject that into the gonad. And the gonad is actually has a common cytoplasm, so the DNA goes in here. It's taken up by the cells, by the germ cells, as they cellularize to become oocytes. They're then fertilized, become babies, and some of those babies will have the transgenic DNA as an extra chromosomal array. And some of those will transmit that array generation after generation, and you get a stable line. Okay, so what you do is you just look for that marker, because you always need to have that control, right? You don't know if what you put in your plasmid is functional or not. Okay, and then you ask, is functional protein expressed from the transgene? Okay? And so the way that we're doing that, we're doing body size. And it's a really simple assay. You just catch the animals on a dissecting scope going forward, because when they reverse, it can affect how their tail looks. You just draw a line through the middle of it, because these animals are really linear. If you take a perimeter shot, then it's like twice going down the center. Anyway, really easy. You have to capture enough worms to make it statistically significant. And then you normalize the population to a control background. In our case, it's a mutant phenotype that we're asking, can the transgene rescue the mutant phenotype? Okay, so we were asking, if we put a transgene that has a mutated variation of LON2 gene in it into animals that don't make their own LON2, will it look more wild type? That's our assay. Okay? So, LON2 animals, now before I normalize to the wild type, here I'm normalizing to the background, which is LON2. This is the particular allele that knocks out the function, okay? Wild type is actually now about 84% LON2, just to give you an idea of what rescuing back to the wild type looks like. Okay, we put in our full-length transgene, just tagged with the GFP, and this rescues animals right back down to wild type, if not better. Okay? And so, now that we've proven that, now we can start to bash it. We can take out everything that we're asking, does it play a role in its function? And I showed you a variety of post-translational modifications that could potentially affect the activity of our protein. And so, the first thing that we asked, because we knew that this heparin sulfation, that's the sugar, that's the particular sugar that's attached to this protein. Is it necessary? for LON2 to inhibit our TGF-beta and the whole line. So we knocked out the 3 sites that the sugar should bind to. It's still rescued fine. Okay, good. So we now know that the core protein is sufficient for this activity. Does it need to be bound to the plasma membrane? And so we took off the GPI link. It's still rescued. We put this back into our animals lacking its own LON2 and asked if this transgene can rescue. It does. So soluble protein can rescue. It doesn't have to be located at the plasma membrane. So I think I forgot to tell you that this is actually expressed on the same cell that the receptors are located at. And so it's physically sequestering. The idea is that this regulator, LON2 glucagon, is sequestering the TGF-beta from the receptors, preventing the receptors from being activated. Okay, how low can you go? Nobody had asked, well, what about this protease site? And so this is the protease site, geez, okay. So we created a plasmid where it had only this much of the gene sequence. And we asked, is that active? Now, one of the things that have been noted in some cancer cell lines is that the shed glypican is higher in some of these cancer cell lines than normally seen. And so, is that just a byproduct, or could that actually be active? Okay? And it can be shed either by losing the GPI, or it can be shed by protease cleavage. And so, we asked, can this protease product contain activity in our system? Yes, it did. Okay? It has no sugar attachment sites. It's perfectly soluble. It is secreted. And it still has this protein-protein interaction site. Now, what nobody in the field had done before, when they looked at the soluble product from the protease cleavage, there are actually 2 parts that are made. Nobody had asked, what about that other part? Is that active? And so in order to do this, we had to put the signal sequence onto here so it actually gets to where it needed to go. And we found this site was active too. We had just shown that the activity resides in this part, right? And so the C-terminal region alone is also active. And so, okay. It doesn't have to, we did put a GFP on it and showed that that was active too. I'm just not showing that here. So we did it 2 different ways and showed that it is active. And so we asked, okay, this one has a couple of the sugars. It has the membrane anchorage. So we took out those 2 sugars, attachment sites. It no longer rescued, showing that for this particular part of the glypican, that the sugars were important. Furthermore, when we took off the GPI link, see here we did it in the context of the GPI, the GFP, the green fluorescent protein, because we showed it worked. Again, it did not rescue, even though it had the sugar attachment sites. So this one's a more complicated story. So we've shown that this part rescues, that this part is functional by itself, but this part is functional too. But this one requires its sugar attachment sites, and it needs to be attached to the membrane. So there is something special about membrane localization. Not necessary, but sufficient. Okay, I mentioned that there were how many glypicans in C. elegans? Play with me here. 3. Yeah. Okay, it's 2. It's 2. And so the question becomes, okay, is heparin sulfation of a protein sufficient? This particular glypican is not expressed in the same time or place. And so if we put it in our LON2 mutant worms, using LON2's own promoter, so this other glypican is being expressed at the same time in the same place as a LON2 actually work? Can that be functional? And this one even has 4 sugar attachment sites. But the answer is no. But it actually has a GPI link. It has the sugars, which we saw by themselves were functional. And so what is that telling us? You guys are tough. Okay. It's saying that the heparin sulfate and being it localized to the right place isn't sufficient. But there's something else going on that's lending specificity to this interaction. Okay, so let's go back to the protein core, this minimal core region that we discovered has activity, but it has this protein-protein interaction motif. So what we did is we did a deletion of this part because it's only 12 amino acids from here to there. And then we also did amino acid substitution here. And what we found that this site was absolutely critical, suggesting that there was a factor X involved, which is really cool. We had previously shown that this is not required in the context of the full length. But what does the full length protein also have? The full length protein, I hear sugar attachment sites. Yes, it has sugar attachment sites, which this new work showed can independently have activity. And so what we did is we took out the sugar attachment sites, and we took out this, and we asked if it was still functional. It was. Okay. Still has the membrane anchorage, which we know is important for that little C-terminal portion. So fine. We bashed this even further. We took off the membrane attachment site, showed that now this is no longer functional. So there's something special going on here. Something special. Can you hear me? Because I'm fluctuating. Good. So what LON2 needs, this glycogen needs, is either attachment to the cell membrane or the RGD that is a protein-protein interaction motif. But neither of them is critical. Either or. It works. Okay, and so that made us think maybe it's interacting in a complex with something. And so, and there was this long disordered region that had the sugar side chain, so maybe its location at the membrane might be important. If you think about the cell, and you think, okay, LON2 is up here, the interaction motif is way up here, maybe it's interacting with the transmembrane domain protein, something that will keep it linked to the cell at the cell surface. And so what we did is we just took out this linker region and affixed a GPI link to it and asked, does that affect its activity? And it worked fine. So apparently the 3D stoichiometry, or not stoichiometry, the 3D presentation of it on a cell surface is not that critical, or else it's just flexible enough there that it can still be functional. Okay. And since we hooked everything up to GFP, I can show you where it localizes to cell-cell, to the cell surface, just right where you'd expect it to, right? Because it has the GPI anchor, right? And so it's a little bit different when you use that small part for the GPI anchor. It's still there. It looks like it might be not getting out of the cell quite as well. But enough of it is there. And remember, this one rescues. Okay. So in summary, yeah, okay. So now we have 2 parts that can independently inhibit signaling. One of them is the N-terminal protein core that contains this protein-protein interaction motif. And the second is the C-terminal portion of this molecule. But this one requires the sugar attachment sites, post-translational modifications, plus attachment to the extracellular plasmid. Okay. So I was hoping that I could find a nice little peptide region, nice and druggable, and I found out that it's actually much more complicated than previously appreciated. But also, it might tell us that, it tells us that this part is functional. And so if you're looking at cancer cell lines that have a lot of shed protein, that it may actually be doing something and not just be a byproduct. Also that we need to pay attention to this part of the protein as well in future studies in this and other systems. So we're going to narrow. What we also found out is that activity lies in that core protein beyond just that protein-protein interaction motif. That interaction motif is not what's actually interacting with the TGF-beta. Because remember, that full-length protein was active without it. So that means that there's another region of this protein that is actually interacting with our TGF-beta. Okay, we're going to find out what that is. And then we're going to do more